The big issue with pain is what the go-to medicines, so the prescribed medicines at the moment are opioids and gabapentinoids, and they're both associated with some serious side effects. So with opioids you get itch, you get constipation, you get tolerance and addiction, and a lot of people have heard obviously about the opioid crisis in the US, where there's a lot of abuse of opioids, and it's a really chilling statistic that I use in my talk, that up to 80 to 100 people die of opioid-related deaths per day in the US, which is massive. And with gabapentinoids, not nearly as serious as that, but they do have some significant side effects, and a lot of patients can't keep taking the medicines. They're both very powerful analgesics, but they're not clean. So the desire here is to get non-opioid painkillers, and ion channels are attractive, for example in the case of opioids, because there's an ion channel that's downstream of the opioid receptor, like calcium channels. So instead of using opioids, we can try and hijack or short-circuit that signaling mechanism and go and modulate the ion channels directly, so you can get analgesia without addiction and side effects. So people have been looking at calcium channels and sodium channels and TRP channels that are in the sensory neurons, but they're not associated with the opioid receptors. So trying to get more safe and effective medicines that patients will want to take. A lot of people would say that ion channels are a more difficult target class to prosecute. I don't know whether the facts actually support that. I think there's some issues with ion channels that are shared with other target classes. For example, trying to get drug-like molecules. Some molecules have gone to the clinic that are not very drug-like, and so they failed. They might have tox, they might have poor PK. So the challenge with ion channels is to get drug-like molecules, but I think that's a general challenge for drug discovery. Specifically for ion channels, the pain targets of interest are part of larger gene families, so sodium channels and calcium channels. They're closely related proteins, and so a compound quite often will hit or bind to multiple ion channels in that family, and so you get side effects that way. So there's been a big search for selectivity, people looking for novel binding sites, and the most popular story is NaV1.7. So Pfizer and other pharmaceutical companies discovered a novel binding site which gives very potent compounds that are very selective for a particular ion channel of interest. So that's the big search now, is for novel binding sites with small molecule approaches. And I think some of the people who are making biologics as well, antibodies, they hope that those biological ligands will get more specificity than small molecules. They'll be able to ferret out small binding pockets in the ion channels of interest without hitting the gene family members. So they'll be more selective, and they'll be more safer with fewer side effects. Right, this was, I've talked about an 8-year collaboration we have with Grunenthal. So we looked at 5 lead series, and at the end of 8 years we have a formal lead series that's done IND toxicology, and we also have a backup series. So these have been patented, these are being out-licensed, so that was a successful effort. Again, 8 years, quite a long time, but that's fairly typical for drug discovery. But on a personal and professional level, this was a true collaboration. We're a CRO, we don't always get to talk about the work, we sometimes just get asked to deliver the data. But we had a really nice ongoing discussion, two-way discussion with Grunenthal. They viewed us as an equal collaborator. So we have very good professional and some personal relationships with that. And that's very rewarding as a CRO. Sometimes we have very transactional relationships with clients, but with these bigger projects we build more of a relationship. So the people who worked on this project definitely took away that positive from the work we did.